The lymphatic system is part of the circulatory system, comprising a network of lymphatic vessels that carry a clear fluid called lymph, directionally towards the heart. The lymphatic system was first described in the 17th century independently by Olaus, Rudbeck and Thomas Bartholin. Unlike the cardiovascular system, the lymphatic system is not a closed system. The circulatory system processes an average of 20 liters of blood per day through capillary filtration, which removes plasma while leaving the blood cells. Roughly 17 liters of the filtered plasma get reabsorbed directly into the blood vessels, while the remaining 3 liters are left behind in the interstitial fluid. One of the main functions of the lymph system is to provide an accessory route for these excess 3 liters per day to get returned to the blood. The other main function is that of defense in the immune system. Lymph is very similar to blood plasma but contains lymphocytes and other white blood cells. It also contains waste products and debris of cells together with bacteria and protein. Associated organs composed of lymphoid tissue are the sites of lymphocyte production. Lymphocytes are concentrated in the lymph nodes. The spleen and the thymus are also lymphoid organs of the immune system. The tonsils are lymphoid organs that are also associated with the digestive system. Lymphoid tissues contain lymphocytes, and also contain other types of cells for support. The system also includes all the structures dedicated to the circulation and production of lymphocytes, which also includes the bone marrow, and the lymphoid tissue associated with the digestive system. The blood does not come into direct contact with the parenchymal cells and tissues in the body, but constituents of the blood first exit the microvascular exchange blood vessels to become interstitial fluid, which comes into contact with the parenchymal cells of the body. Lymph is the fluid that is formed when interstitial fluid enters the initial lymphatic vessels of the lymphatic system. The lymph is then moved along the lymphatic vessel network by either intrinsic contractions of the lymphatic passages or by extrinsic compression of the lymphatic vessels via external tissue forces, or by lymph hearts in some animals. The organization of lymph nodes and drainage follows the organization of the body into external and internal regions. Therefore, the lymphatic drainage of the head, limbs, and body cavity walls follows an external route, and the lymphatic drainage of the thorax, abdomen, and pelvic cavities follows an internal route. Eventually, the lymph vessels empty into the lymphatic ducts, which drain into one of the 2 subclavian veins. Structure. The lymphatic system consists of lymphatic organs, a conducting network of lymphatic vessels, and the circulating lymph. Lymphoid tissue is divided into primary, secondary, or tertiary depending on the stage of lymphocyte development involved. The tertiary lymphoid tissue typically contains far fewer lymphocytes, and assumes an immune role only when challenged with antigens that result in inflammation. It achieves this by importing the lymphocytes from blood and lymph. The thymus and the bone marrow constitute the primary lymphoid organs involved in the production and early selection of lymphocyte tissues. Bone marrow is responsible for the creation of both T cells, and the production and maturation of B cells. From the bone marrow, B cells immediately join the blood system and travel to secondary lymphoid organs in search of pathogens. T cells on the other hand travel from the bone marrow to the thymus where they are allowed to develop further. Mature T cells join B cells in search of pathogens. The other 95% of T cells begin a process of apoptosis. The central or primary lymphoid organs generate lymphocytes from immature progenitor cells. Secondary or peripheral lymphoid organs, which include lymph nodes and the spleen, maintain mature naive lymphocytes and initiate an adaptive immune response. The peripheral lymphoid organs are the sites of lymphocyte activation by antigen. Activation leads to clonal expansion and affinity maturation. Mature lymphocytes recirculate between the blood and the peripheral lymphoid organs until they encounter their specific antigen. Secondary lymphoid tissue provides the environment for the foreign or altered native molecules to interact with the lymphocytes. It is exemplified by the lymph nodes, and the lymphoid follicles in tonsils, Peyer's patches, spleen, adenoids, skin, etc. that are associated with the mucosa-associated lymphoid tissue. Thymus. The thymus is a primary lymphoid organ and is the site of the T cells, the lymphocytes of the adaptive immune system. Spleen. Lymph nodes. A lymph node is an organized collection of lymphoid tissue, through which the lymph passes on its way back to the blood. Lymph nodes are located at intervals along the lymphatic system. Several afferent lymph vessels bring in lymph, which percolates through the substance of the lymph node, and is then drained out by an efferent lymph vessel. The substance of a lymph node consists of lymphoid follicles in an outer portion called the cortex. The inner portion of the node is called the medulla, which is surrounded by the cortex on all sides except for a portion known as the hilum. The hilum presents as a depression on the surface of the lymph node, which makes the otherwise spherical lymph node, bean-shaped or ovoid. The efferent lymph vessel directly emerges from the lymph node here. The arteries and veins supplying the lymph node with blood enter and exit through the hilum. A lymph follicle is a dense collection of lymphocytes, the number, size and configuration of which change in accordance with the functional state of the lymph node. For example, the follicles expand significantly when encountering a foreign antigen. The selection of B cells, also called B lymphocytes, occurs in the germinal center of the lymph nodes. Lymph nodes are particularly numerous in the mediastinum in the chest, neck, pelvis, axilla, inguinal region, and in association with the blood vessels of the intestines. Other lymphoid tissue associated with the lymphatic system is concerned with immune functions in defending the body against infections and the spread of tumors. It consists of connective tissue with various types of leukocytes, mostly lymphocytes enmeshed in it, through which the lymph passes. Regions of the lymphoid tissue that are densely packed with lymphocytes are known as lymphoid follicles. Lymphoid tissue can either be structurally well organized as lymph nodes or may consist of loosely organized lymphoid follicles known as the mucosa-associated lymphoid tissue. Lymphatics. Lymphatics refer to the vessels which conduct lymph between different parts of the body. These tubular vessels include the lymph capillaries, the lymph vessels, the right lymphatic duct and the thoracic duct, also called the left lymphatic vessel. Tubular vessels transport back lymph to the blood, ultimately replacing the volume lost from the blood during the formation of the interstitial fluid. These channels are the lymphatic channels, or simply called lymphatics. Development. Lymphatic tissues begin to develop by the end of the 5th week of embryonic development. Lymphatic vessels develop from lymph sacs that arise from developing veins, which are derived from mesoderm. The first lymph sacs to appear are the paired jugular lymph sacs at the junction of the internal jugular and subclavian veins. From the jugular lymph sacs, lymphatic capillary plexuses spread to the thorax, upper limbs, neck and head. Some of the plexuses enlarge and form lymphatic vessels in their respective regions. Each jugular lymph sac retains at least one connection with its jugular vein, the left one developing into the superior portion of the thoracic duct. The next lymph sac to appear is the unpaired retroperitoneal lymph sac at the root of the mesentery of the intestine. It develops from the primitive vena cava and mesonephric veins. Capillary plexuses and lymphatic vessels spread from the retroperitoneal lymph sac to the abdominal viscera and diaphragm. The sac establishes connections with the cisterna chali but loses its connections with neighboring veins. The last of the lymph sacs, the paired posterior lymph sacs, develop from the iliac veins. The posterior lymph sacs produce capillary plexuses and lymphatic vessels of the abdominal wall, pelvic region, and lower limbs. The posterior lymph sacs join the cisterna chali and lose their connections with adjacent veins. With the exception of the anterior part of the sac from which the cisterna chali develops, all lymph sacs become invaded by mesenchymal cells and are converted into groups of lymph nodes. The spleen develops from mesenchymal cells between layers of the dorsal mesentery of the stomach. The thymus arises as an outgrowth of the third pharyngeal pouch. Function. The lymphatic system has multiple interrelated functions. It is responsible for the removal of interstitial fluid from tissues, it absorbs and transports fatty acids and fats as chyle from the digestive system, it transports white blood cells to and from the lymph nodes into the bones, the lymph transports antigen-presenting cells, such as dendritic cells, to the lymph nodes where an immune response is stimulated. Function of the fatty acid transport system. Lymph vessels called lacteals are present in the lining of the gastrointestinal tract, predominantly in the small intestine. While most other nutrients absorbed by the small intestine are passed onto the portal venous system to drain via the portal vein into the liver for processing, fats are passed onto the lymphatic system to be transported to the blood circulation via the thoracic duct. Are fatty acid esters of glycerol that passively diffuse from the GI tract to the portal system. The enriched lymph originating in the lymphatics of the small intestine is called chyle. The nutrients that are released to the circulatory system are processed by the liver, having passed through the systemic circulation. Immune function. Clinical significance. The study of lymphatic drainage of various organs is important in diagnosis, prognosis, and treatment of cancer. The lymphatic system, because of its physical proximity to many tissues of the body, is responsible for carrying cancerous cells between the various parts of the body in a process called metastasis. The intervening lymph nodes can trap the cancer cells. If they are not successful in destroying the cancer cells, the nodes may become sites of secondary tumors. Lymphadenopathy. Lymphadenopathy refers to one or more enlarged lymph nodes. Small groups or individually enlarged lymph nodes are generally reactive in response to infection or inflammation. This is called local lymphadenopathy. When many lymph nodes in different areas of the body are involved, this is called generalized lymphadenopathy. Generalized lymphadenopathy may be caused by infections such as infectious mononucleosis, tuberculosis and HIV, connective tissue diseases such as SLE and rheumatoid arthritis, and cancers, including both cancers of tissue within lymph nodes, discussed below, and metastasis of cancerous cells from other parts of the body, that have arrived via the lymphatic system. Lymphedema. Lymphedema is the swelling caused by the accumulation of lymph, which may occur if the lymphatic system is damaged or has malformations. It usually affects limbs, though the face, neck and abdomen may also be affected. In an extreme state, called elephantiasis, the edema progresses to the extent that the skin becomes thick with an appearance similar to the skin on elephant limbs. Causes are unknown in most cases, but sometimes the patient in question has a previous history of severe infection, most commonly caused by a parasitic disease, like the lymphatic filariasis for example. Lymphangiomatosis is a disease involving multiple cysts or lesions formed from lymphatic vessels. Lymphedema can also occur after surgical removal of cancerous lymph nodes in the armpit or groin. Treatment is done by massage, and is not permanent. Cancer. Cancer of the lymphatic system can be primary or secondary. Lymphoma refers to cancer that arises from lymphatic tissue. Lymphoid leukemias and lymphomas are now considered to be tumors of the same type of cell lineage. They are called leukemia when in the blood or marrow and lymphoma, when in lymphatic tissue. They are grouped together under the name lymphoid malignancy. Lymphoma is generally considered as Hodgkin's lymphoma and non-Hodgkin's lymphoma. Hodgkin's lymphoma is characterized by a particular type of cell, called a Reeder-Uro-Sternberg cell, visible under microscope. It is associated with past infection with the Epstein-Barr virus, and generally causes a painless rubbery lymphadenopathy. It is staged, using Ann Arbor staging. Chemotherapy generally involves the ABVD and may also involve radiotherapy. Non-Hodgkin's lymphoma is a cancer characterized by increased proliferation of B cells or T cells, generally occurs in an older age group than Hodgkin's lymphoma. It is treated according to whether it is high-grade or low-grade, and carries a poorer prognosis than Hodgkin's lymphoma. Lymphangiosarcoma is a malignant soft tissue tumor, whereas lymphangioma is a benign tumor occurring frequently in association with Turner syndrome. Lymphangioliomyomatosis is a benign tumor of the smooth muscles of the lymphatics that occurs in the lungs. Lymphatico-venous communications. Present research has found clues about a lymphatico-venous communication. In mammals, lymphatico-venous communications other than those at the base of the neck are not easy to demonstrate, but described in some experiments. the specialists observed that the pulmonary complications following lymphangiography are more often severe in patients with lymphatic obstruction. In these cases, the contrast medium is thought to reach the vascular system via lymphovenous communications which shunt the material directly into the venous stream, bypassing those lymph nodes distal to the communications. Because less contrast agent is absorbed in lymph nodes, a greater portion of the injected volume passes into the vascular system. Since pulmonary complications are related to the amount of medium reaching the lungs area, the early recognition of lymphovenous communications is of great significance to the lymphangiographer. Another hint in proving a lymph vein communication is offered by a Robert F. Dunn experiment. The passage of radioactively tagged tracers, injected at elevated pressure, through the lymph node venous communications coincides with the increased pressures of injection and subsequent nodal palpation in dogs. The passage of iodinated I-125 serum albumin indicates that direct lymph node venous communications are present, whereas passage of nucleated erythrocytes requires a communication structure the size of a capillary or larger. Moreover, the evidence suggests that in mammals under normal conditions, most of the lymph is returned to the bloodstream through the lymphatico-venous communications at the base of the neck. When the thoracic duct venous communication is blocked, however, the resultant raised intralymphatic pressure will usually cause other normal non-functioning communications to open and thereby allow the return of lymph to the bloodstream. History: Hippocrates was one of the first people to mention the lymphatic system in 5th century BC. In his work on joints, he briefly mentioned the lymph nodes in one sentence. Rufus of Ephesus, a Roman physician, identified the axillary, inguinal and mesenteric lymph nodes as well as the thymus during the 1st to 2nd century AD. The first mention of lymphatic vessels was in 3rd century BC by Herophilos, a Greek anatomist living in Alexandria, who incorrectly concluded that the absorptive veins of the lymphatics, by which he meant the lacteals, drained into the hepatic portal veins, and thus into the liver. The findings of Rufus and Herophilos were further propagated by the Greek physician Galen, who described the lacteals and mesenteric lymph nodes which he observed in his dissection of apes and pigs in the 2nd century AD. In the mid-16th century, Gabriele Falloppio described what are now known as the lacteals as coursing over the intestines full of yellow matter. In about 1563 Bartolomeo Eustachi, a professor of anatomy, described the thoracic duct in horses as vena alba thoracis. The next breakthrough came when in 1622 a physician, Gaspar Aselli, identified lymphatic vessels of the intestines in dogs and termed them venae albae lactei, which is now known as simply the lacteals. The lacteals were termed the fourth kind of vessels, and disproved Galen's assertion that chyle was carried by the veins. But, he still believed that the lacteals carried the chyle to the liver. He also identified the thoracic duct but failed to notice its connection with the lacteals. This connection was established by Jean Pecquets in 1651, who found a white fluid mixing with blood in a dog's heart. He suspected that fluid to be chyle as its flow increased when abdominal pressure was applied. He traced this fluid to the thoracic duct, which he then followed to a chyle-filled sac he called the chyli receptaculum, which is now known as the cisternae chyli. Further investigations led him to find that lacteals' contents enter the venous system via the thoracic duct. Thus, it was proven convincingly that the lacteals did not terminate in the liver, thus disproving Galen's second idea, that the chyle flowed to the liver. Johann Vislingius drew the earliest sketches of the lacteals in humans in 1647. The idea that blood recirculates through the body rather than being produced anew by the liver and the heart was first accepted as a result of works of William Harvey, a work he published in 1628. In 1652, Olaus Rudbeck, a Swede, discovered certain transparent vessels in the liver that contain clear fluid, and thus named them hepatico-aqueous vessels. He also learned that they emptied into the thoracic duct, and that they had valves. He announced his findings in the court of Queen Christina of Sweden, but did not publish his findings for a year, and in the interim similar findings were published by Thomas Bartholin, who additionally published that such vessels are present everywhere in the body, and not just the liver. He is also the one to have named them lymphatic vessels. This had resulted in a bitter dispute between one of Bartholin's pupils, Martin Bogdan, and Rudbeck, whom he accused of plagiarism. Galen's ideas prevailed in medicine until the 17th century. It was believed that blood was produced by the liver from chyle contaminated with ailments by the intestine and stomach, to which various spirits were added by other organs, and that this blood was consumed by all the organs of the body. This theory required that the blood be consumed and produced many times over. Even in the 17th century, his ideas were defended by some physicians. Etymology: the adjective used for the lymph-transporting system is lymphatic. The adjective used for the tissues where lymphocytes are formed is lymphoid. Lymphatic comes from the Latin word lymphaticus, meaning connected to water. See also, List of lymphatic vessels of the human body, American Society of Lymphology, lymphatic system, equivalent for the central nervous system, lymphangiogenesis, lymphangin, lymphatic system assessment, manual lymphatic drainage, mononucleophagocyte system, Waldemar-Olszewski. Discovered fundamental processes in human tissues connected with function of the lymphatic system, Ru. Th microwatt micron th micron th micron th 1/2 th 3/4 th micron th 1/2 th 1/2 th degree th to the first th degree th 1/4 thn euro th 3/4 th squared. THNA author of the lymphatic system segmental theory, Kikuchi disease, references. External links, lymphatic system overview.